Good morning, everyone. Good morning. Good morning. Good morning, all. Good morning. Good morning. Okay, so friends, in the last class, we have completely understand the translation in case of a prokaryotic organisms. Now, what we have to read, friends? Now we have to read the process of translation in case of a eukaryotic organism. So friends, if you quickly recall the differences between the process of translation is obviously same whether it is a prokaryotic organism or whether it is a eukaryotic organism, doesn't matter, friends. The process of translation is absolutely same. There is a no difference at all. So but the process is same, but the type of factors involved is different. Good morning, everyone. Good morning. The process of, the factors involved is different, and there are certain fundamental differences also there between the prokaryotes and eukaryotes. Because, friends, we are very complicated organisms when compared to the prokaryotic organism. So, what I can say, in prokaryotic organism, process is simple. In case of eukaryotic organism, process is at most complex. Friends, one of the most important or the fundamental difference that I want you people to remember is that if you quickly recall, in case of a prokaryotic organism, whatever the first tRNA comes, tRNA is bringing the methionine, and if you all remember, friends, that methionine is basically added by the pharmyl group, is it? So that's why, friends, we basically are generally called it as the F methionine or F met tRNA, right? But friends, in case of a eukaryotic organism, once again listen to me carefully, we can call it as the F met tRNA in case of prokaryotes. But friends, in case of a eukaryotic organism, we never ever call it as the F met tRNA. We generally call it as what, friends? Amino acyl tRNA, which means amino acid plus tRNA, that's all. And second very important point is what, in case of a prokaryotic organism, we people read very clearly, there is a presence of Shine and Dalgarno sequence. That is what we studied. But friends, when you look into the eukaryotic organism, there is a no presence of the Shine and Dalgarno sequence. Instead of the Shine and Dalgarno sequence, in case of eukaryotes, what is present, friends? In case of eukaryotic organism, Kojak sequence will be present. Okay, these are all some of the fundamental differences between the prokaryotic and eukaryotic, but the process is easy, process is same. But friends, the type of factors, that is the initiation factors, elongation factors, and termination factors that are involved is entirely different. All of you pay attention, friends, it's easy to understand. So there are three type of mRNAs, that's a common. So friends, listen to me for the period of 15 minutes continuously. Okay, same like a prokaryotic organism, here also, friends, we require three type of RNAs. One we can call mRNA, tRNA, rRNA. We all commonly know that all the three RNAs are compulsory required for the process of translation or protein synthesis. mRNA is the one which brings the information. mRNA always bring the information what? In the form of codons. We all commonly know that what the tRNA is doing. tRNA is the one which brings the amino acid. That's why tRNA we can call the adapter tRNA. I hope you remember, friends. In the prokaryotic organism, we studied very specially how the tRNA is getting activated. Same way here also. mRNA, tRNA, then friends rRNA. mRNA bring the information, tRNA bring the amino acid. By using this particular amino acid and the information present on the mRNA, friends, there will be a synthesis of proteins. Very simple. Same in case of prokaryotes, we read that initiation, elongation and termination factors. Same way, friends, in case of eukaryotics also, initiation factors, elongation factors and termination factors. But friends, the number of factors involved is more when compared to the prokaryotes. That's the only difficulty here. Same, what do we require? Undoubtedly we require this particular ribosome complex. I told you, right, friends, in the large ribosomal subunit what enzyme is present? Hope every one of you remember the most important enzyme. What do you call it as, friends? Peptidyl transferase enzyme will be present. Peptidyl transferase enzyme can also called as what, friends? Catalytic RNA. We can also call it as what, friends? Ribozyme. Kindly don't forget that. Same, right? There are three sites are present in the ribosome complex. One we called E site, P site, A site. I have very clearly mentioned in the E site what kind of tRNAs basically enter. We read very clearly, friends. In the E site, whatever the tRNA that is uncharged. I hope every student remember. In the prokaryotic translation we studied very clearly when we can called a tRNA is the uncharged tRNA. We can call a tRNA is the uncharged tRNA whenever it is not associated with this particular amino acid. Then only it can exit. P site is what? The most important site where the peptide bond is getting formed. And friends, what is A site? A site is the area where the amino acid with the tRNA will come and associate. Very simple point. In the last class, we studied the prokaryotic subunits, and I told to all of you exclusively to remember the subunits, right? Remembering subunits are very important. We read 30s contain 16s, 50s contain 23s, 5s. Okay, same way. Compare with the prokaryotic organism. By that, friends, you can remember very easily eukaryotic organism. The 40s subunit basically contain what, friends? 18S rRNA. Whereas, friends, what does this particular 60S consist of? It consists of 28S, it consists of 5.8S and it consists of 5S. So friends, I told you why I am saying to remember the small subunits are because every small subunit having their own significance. Every small subunit having their own importance. That's why it is necessary to remember every single subunit without fail. Okay, so same, let's try to understand elaboratively the differences between the initiation, elongation as well as the termination. Friends, every single step try to compare with the prokaryotic organism. By that, friends, you can remember for a very long time. If every one of you quickly recall, in case of a prokaryotes, we all remember hardly, friends, there is an involvement of three factors. That's all. Hardly three factors are there. One we call it as what? IF1 we read. Another one we call it as IF2 GTP molecule we studied, friends. Another one we studied, friends, IF3. That's all. Only there is an involvement of just three factors. That's all. But friends, in case of eukaryotes, there are many factors. Now you only try to look into this. I will be explaining everything in a simple way. EIF2, 2, 2B, 3, 4A, 4B, 4E, 4G, 5 and 6. There are many factors are associated in the eukaryotes. Friends, what is the best way to remember every single factor? The best way to remember the single factor is comparing with the prokaryotic translation. It's very very easy. Okay, every one of you please listen my statements carefully, friends. What we have to do actually in the initiation, I have told you all of you clearly. In the initiation, everyone listen to me, friends. In the initiation, we have to assemble everything. We have to assemble mRNA, we have to assemble tRNA. At the same time, friends, we have to assemble the RNA. All these three types of RNA, friends, we have to assemble in the process of initiation. Every students just listen to me very carefully, friends. Listen to me for 10 minutes without interruption. In case of pay attention, in case of eukaryotes, their assembly occur in different ways. What do you mean by the word their assembly occur in different ways? That is, first, mRNA can be assembled separately, friends. Assembly of mRNA is separate. Assembly of tRNA and rRNA is also separate. Once mRNA assembled and tRNA, rRNA assembled separately, all three will assemble with each other. By that, active initiation complex will be formed. Okay, friends, please kindly don't get confused. Just listen my every single word very briefly. I will explain twice, you will understand it in a simple way. Okay, what are the first two factors that are associated with the small ribosomal subunit? Every one of you please remember, this is the small ribosomal subunit 40S. So friends, every one of you please listen. The small subunit of the 40S can be associated with the EIF1A and EIF3. These two factors associated with the small ribosomal subunit, that is the 40S. And friends, listen to me very carefully. Who will be associated with the large ribosomal subunit, friends? Large ribosomal subunit can be associated with the EIF5 followed by, friends, EIF6. EIF5, EIF6, both are associated with each other. Now listen to me carefully and keep on comparing. Friends, focus. In case of a prokaryotic organism, what is the name of the initiation factor that brings the first tRNA? I hope every one of you remember. The first tRNA can be brought by the IF2 GTP molecule. In case of a prokaryotes, friends, don't get confused. Here also same. We have to just add the letter E. EIF2 GTP. This is EIF2 GTP. This is the one, friends, who is going to bring this particular methionine tRNA. Now friends, you only try to understand the concept very clearly. Do you think, is there any formation of the formyl methionine tRNA? No, right, friends? Every student listen to me carefully, friends. Look into the diagram. There is a tRNA associated with the amino acid methionine, then followed by, friends, EIF2 GTP molecule. So in a simple way, there is a formation of a tertiary complex consists of a tRNA amino acid, then followed by the EIF2 GTP. Very, very, very simple point. Okay, now what all got assembled? Every student please listen to me. What got assembled? 30S ribosomal subunit is assembled, and tRNA amino acid is also assembled. Once again, I am repeating quickly. Look into this. First, just listen. Small ribosomal subunit can be associated with the EIF1A and 3. Later we will read large ribosomal subunit can be associated by the EIF5 and EIF6. Don't confuse, it's very, very easy. And Who is going to bring the first tRNA, which is only methionine tRNA? It is not at all added by the pharmyl group. That can be associated by the EIF2 GTP molecule. This is fine. Everyone look into this. This is the simplified complex which contains small ribosomal subunit, initiation factors and tRNA. Friends, now everyone listen. We have to assemble the mRNA also, right? mRNA is the one which is having the entire information. Before understanding how does this particular mRNA can be assembled, every one of you please listen to me very carefully, friends. This particular mRNA can be associated with a complex. We call it, friends, EIF4F complex. What is that? EIF4F complex. What is this particular EIF4F complex consist of? This 4F complex consists of E, G, A. And every students, please kindly remember, friends, if the A wants to be function, don't confuse. You have to remember this. The moment you understand once or twice, the concept will be very clear. The A can be associated with what, friends? A can be associated with the B and H. Very, very simple point. And friends, every one of you listen to me very carefully. Here there is a presence of G, right? And friends, what I can call the protein G? I can call it the protein G as the scaffold protein. What I can call, friends, G is the scaffold protein. Friends, what is the function of a scaffold protein? We all know, right? Scaffold protein is a G which can hold the E and A together. Very simple point. And what I told to all of you, if the A wants to occur, it requires the help of B and H. So this is how, friends, this entire EIF4F complex associated with the mRNA. That is absolutely fine. So friends, now 30S ribosomal subunit with the tRNA is ready. Same way mRNA is ready, but friends, tRNA, small RNA and mRNA associated differently. I mean to say separately. But friends, the concept is very clear. If I want to form the, every one of you listen, if I want to form the active initiation complex, what do I need? In order to form the active initiation complex, I compulsory need small rRNA, tRNA and mRNA together. Now, how this assembly occur? And every one of you listen my statements very carefully, friends. Near the AUG in the mRNA, there is a specialized sequence is present which we can call the Kojak sequence based on the name of the discoverer. Okay, now look into the picture. Every one of you try to look into this. It's easy. Now friends, everyone look into this. This is what? Ribosome, small ribosome and tRNA complex. Friends, now this particular small ribosome and tRNA, you can look into the picture very carefully. They start scanning the mRNA. Now you can ask me, sir, I understand it is scanning the mRNA, but how it enters into the mRNA? Of course, friends, with the help of this particular EIF4F complex only we seen it. Now everyone listen, with the help of EIF4F complex, the small ribosomal subunit and the tRNA start scanning the mRNA. Don't forget that, friends. They start scanning the mRNA. Okay, they start scanning the mRNA till what extent? Every one of you listen. Till tRNA anticodon associated with the codon of the mRNA. Very, very simple point it is. Now, small ribosomal subunit is ready, absolutely ready, and mRNA is absolutely ready. tRNA is also absolutely ready. Then friends, what else we required? We require the large ribosomal subunit also compulsory. who is going to bring this particular large ribosomal subunit, friends? The large ribosomal subunit can be brought by EIF5 and EIF6. These are all the two subunits will bring the large ribosomal subunit to this particular complex. Every student has to listen to me carefully. The moment 60S will come here, immediately what is going to happen, friends? All factors can be released out. That is first EIF4F complex, which means which is associated with the mRNA. Everything will get dissociated. And EIF5 and 6 brought what? rRNA, large ribosomal, that also can get dissociated. And very important one, who brought the first tRNA? EIF2 GTP. Friends, that EIF2 GTP undergo the hydrolysis and it can be released in the form of a EIF2 GDP molecule. That's a very simple statement I want you people to remember. In 1 minute I will summarize the entire prokaryotic translation initiation process. Just quickly try to understand it. I think most of the people are sleeping. They went back. Come on, be active. Okay, so this is called the 80S ribosomal complex. 80S ribosomal complex contains 40S and 60S. 40S initially can be associated by the EIF1A followed by the EIF3. Friends, EIF1A and 1,3 associate the 30S and EIF2 GTP will bring this particular methionine tRNA, which we call tertiary complex. Now this entire complex, everyone listen, entire complex associated with the small ribosomal subunit. As I already told you, the entire mRNA is associated with the EIF4F complex. Now friends, the small ribosomal subunit associated with the mRNA and it's keep on scanning the mRNA. Till what extent it's keep on scanning the mRNA? Till it reach the step called as AUG. Please kindly don't forget this particular statement also, friends. Now later I want every student has to understand, the moment the entire complex get established, immediately what is going to happen, friends? 60S ribosomal will come. This can be brought by EIF5 and EIF6. Very simple. Then friends, we formed active initiation complex. I hope everyone understand it very clearly. This is what active initiation complex. Next one, elongation. Of course, friends, which is the most toughest step in the entire protein synthesis? Obviously, elongation only. I will tell to all of you, friends, the elongation will occur in the 3 steps. The same 3 steps we read in the prokaryotic organism also. Friends, here you know, right? In this condition, which sites become free, friends? A site become free, friends. E site is also become free. And we all people know very well where the new tRNA will comes into the picture. Friends, new tRNA with the amino acid, obviously they will comes to which site, friends? A site only. So every student listen, 3 steps will occur in the elongation. The first step, everyone listen to me. The first step is addition of the another amino acyl tRNA to the A site. That is the first step. Second and the most important step, formation of a peptide bond. And third, another very important step, we called it as, friends, translocation. That is moving the ribosome 1 codon ahead. That is what exactly we call it as translocation. So friends, these 3 steps has to occur compulsory during the process of elongation. So let's try to understand 3 steps elaboratively. All the students, please kindly look into it, and I am requesting all of you, please try to compare with the prokaryotic organism. If you start comparing with the prokaryotic organism, it become extremely easy. Everyone listen friends, look into the diagram. When you look into the diagram, focus. What do you call it this as? We will be calling this as the active initiation complex. There is no doubt about it. Now which site is become free? A site. Now immediately friends, immediately you have to compare. In the prokaryotic organism, what is the name of the elongation factor that brings the second tRNA? I hope all of you remember friends. Which one we call it as a EFTU GTP molecule. Absolutely true point. Is it? EFTU GTP is the molecule which brings the tRNA towards the A site. That's a simple point. But friends, same here in case of a eukaryotic organism, please listen to me. The name of the elongation factor that is involved in bringing the tRNA to the A site. What do you call it this friends? We will be calling it as the EF1 alpha GTP. What is that friends? EF1 alpha GTP is the factor which is associated with the second tRNA with the amino acid. Everyone listen. And this EF1 alpha GTP bring the second tRNA to which site? A site. That is the first step, addition of the amino acid tRNA. Now friends, every one of you listen to me carefully. The moment EAF1F GTP associated here, immediately what is happening friends? GTP hydrolysis occur. Immediately friends, EAF1 alpha converted from GTP form to the GDP form. Please kindly do not forget this particular statement at all friends. Now every one of you listen to me very carefully friends. Okay, this step is occur, that is addition of the amino acid tRNA is over. Now what is the most important step friends? That is the peptidyl transferase enzyme, formation of the peptide bond. Same like prokaryotic organism, not even a single step difference. Whatever the peptidyl transferase enzyme present on the larger ribosomal subunit, every one of you look into the diagram, it will cut the, what it will cut? It will cut the amino acid present on the tRNA of the P site. Very simple. It will cut the amino acid present on the tRNA of the P site, and it will add this particular amino acid to the tRNA present on the A site. Now friends, here what kind of reaction will occur? I have explained you clearly in case of prokaryotic organism also. Condensation reaction occur, that is a loss of water. Obviously friends, between the two amino acid there is a bond will be formed, CONH bond, which we can call it as the peptide bond. Very simple. Now here comes another last important point. You only try to look into this picture friends. In case of a tRNA which is present at the P site, everyone understand, whatever the tRNA that is present at the P site, do you think it is completely charged tRNA? Not at all, right friends? This particular tRNA which is present, it is completely uncharged tRNA. So friends, undoubtedly this particular uncharged tRNA friends has to come from P site to which site? E site. And friends, what do you call it that particular process? Ribosome translocation. Which means what? What it exactly means? The ribosomes start moving one codon ahead. What my friends? The ribosome starts moving one codon ahead. That is what exactly friends we can call the ribosome translocation. Once again friends, very quickly recall with the prokaryotic organism. I hope all of you remember. In case of prokaryotic organism, who is involved in the translocation process? EFG-GTP molecule. I hope everyone of you remember. Friends, here the name of the factor is what friends? EF2 GTP molecule. Very simple, right? Now EF2 GTP molecule will come and associated with which site? E site. The moment it associated with the E site, immediately what is going to happen? Ribosome starts moving one codon ahead. When the ribosome starts moving one codon ahead, what is happening friends? Understand everyone, whatever the uncharged tRNA is there in the P site, the uncharged tRNA comes to E site. Whatever the charged tRNA with the amino acids present on the P site, it will come to the P site. Now, which is free? Obviously, A site is free. To the A site, once again, who will come and attaches? tRNA will come and attaches. Once again, peptide bond synthesis occur. Once again, translocation occur. And this process will be continued. And till where this particular process will be continued, friends? Till the ribosome reaches the stop codon. The same thing we read in the prokaryotic organism also. So let me quickly summarize the elongation in 1 minute. Why I will all the time repeat is that by that you don't remember, you don't forget it. Listen to me carefully. This is the active initiation complex. There is an important factor called as EIF1 alpha GTP is the important factor which brings the tRNA to which site, friends? A site. Immediately EF1 alpha undergo the GTP hydrolysis, it get dissociated immediately. Later what is going to happen, friends? This particular what we can say that peptidyl transferase enzyme present in the large ribosomal subunit, it will cut the amino acid present on the tRNA to the P site and it will add to the amino acid present on the tRNA of the A site. There is a condensation or loss of water occur, formation of a peptide bond. After formation of the peptide bond, you people can see it very clearly, friends. Whatever the tRNA that is present in the P site, it is extremely inactive or uncharged tRNA. So uncharged tRNA has to move compulsory to the E site. How it is compulsory moving to the E site, friends? With the help of translocation factor. What do you call this particular translocation factor, friends? We will call the EF3 GTP. Always compare with prokaryotes. That is the way of learning here. And where this factor will come and attaches to the E site. As a result, what is going to happen? The uncharged tRNA from the P site, it will come into E site. Whereas, friends, the charged tRNA will come from A site to the P site. Now, which site is becoming absolutely free, friends? A site is becoming absolutely free. Please kindly don't forget the point. Like this, the entire process will be going on. And friends, every student, I hope you all people remember, right? In case of a prokaryotic organism, I hope you all remember, how does the termination occur? In the termination, there are some factors are involved. I hope all of you remember. RF1 is present, RF2 is also present, RF3 is also present, friends, in the prokaryotic organism. I hope you all people remember, friends. RRF also involved in this particular eukaryotic, in case of prokaryotes. These four are major factors. And we read EFG also present, IF3 also present. That's true. But these four are major in case of a prokaryotes. But friends, when you look into the eukaryotic translation termination process, there will be no involvement of that many termination factors. Friends, just there is an involvement of two termination factors, that's all. One we call it as ERF1, another one we called, friends, ERF3. These are the two major releasing factors and termination factor. Every one of you, please listen my statements carefully. Listen to me. ERF31, which is exactly act like a RRF. That is ribosome recycling factor, which is involved in the dissociation of this particular ribosome. And friends, when you look into the ERF1, I want all the students has to remember this. ERF1 Actually, friends, it looks like tRNA. Everyone, please listen. The releasing factor ERF1 itself, it's look like a tRNA, but it always recognizes what, friends? Sorry, it will always recognize the stop codon. Anyway, all the students listen to me. I will explain the process of termination twice. Just listen to me carefully. What I said, friends, there are only two type of termination factors are there. One we called ERF1, another one, friends, we can called ERF3 GTP molecule. Everyone pay attention. All the students look into the diagram. How does the ERF1 looks like? This is the simple structure of the ERF1. So which means which is looking exactly like tRNA. You can see that picture, friends. Now ERF1 looking like a tRNA. What the ERF1 will do? All the students look into this. It will come and associated with the UAA stop codon. Friends, everyone listen. ERF1 having the only function of recognizing the stop codon. That's all. The moment ERF1 recognize the stop codon, immediately what is going to happen, friends? ERF1 recruit the ERF3 GTP. Now everyone pay attention. As I already told to all of you, friends, ERF3 GTP can starts behaving like what, friends? It starts behaving like a RRF. So friends, everyone has to listen to me carefully. So first, what it will do? This RRF, which is a ERF3 GTP, it gives the signal to the peptidyl transferase enzyme. What kind of signal it is giving to the peptidyl transferase enzyme, friends? That is to cleave this peptide bond. By that, friends, entire polypeptide comes out. Now every student has to listen my words. This ERF3 GTP immediately recruits what, friends? It is going to recruit the EF2 GTP molecule. Now you have to recall, sir, why it is recruiting the EF2 GTP molecule. I have clearly mentioned that EF2 GTP is involved in the translocation. Now you only try to understand this particular mRNA is what, friends? It is not mRNA, tRNA. This particular tRNA, after cleaving the polypeptide, it will become inactive, right? Obviously the inactive polypeptide has to come to the inactive tRNA has to come to the E site. How does it will come? Of course, with the help of EF2 GTP. Once it comes to the E site, it will get exit, and peptide chain also we have released out. Now here comes the important point. How can I separate? Good morning, Tarun. How can I separate the large ribosomal subunit and small ribosomal subunit? I hope every student remember. Large ribosomal subunit can brought by what, friends? We read that large ribosomal subunit can be brought by EIF5 and EIF6. These are the two factors which are bringing this particular subunit. Now EIF5 and EIF6 associated with the large ribosomal subunit, friends, and immediately it will cause the dissociation of all. And anyway, they are going to assemble immediately also. Once again, I am summarizing, friends. Every students pay attention. It's a very easy process. Whenever ribosome reaches the stop codon, here what is happening? The ERF1, which is called as a eukaryotic releasing factor 1, whose structure is similar to the tRNA. Whose structure is similar to the tRNA. What the ERF1 will do? It will go and recognize the stop codon. Immediately, friends, immediately ERF1 recruits ERF3 GTP molecule. And what did I tell to all of you? Whatever the functions performed by the RRF in case of a prokaryotic organism, the same functions performed by the ERF3 GTP in the eukaryotes. Everyone listen to me. What is the, understand everyone, what is the first function? performed by the ERF3 GTP in the eukaryotes. The first function is ERF3 GTP give the signal to the peptidyl transferase enzyme. Okay, what kind of a signal it is giving to the peptidyl transferase enzyme to cut the peptide bond? So peptide bond came out, there is no doubt about it. Every students listen to me carefully. Once the peptide bond comes out, whatever the tRNA that are present on the P site, it will actually become uncharged tRNA. Of course, true. And we also know that that uncharged tRNA has to come from P site to the E site. Very simple, right? Now, who will do this? This can be done by what we discussed, friends. EF2 GTP molecule. What is that? One second. EF2 GTP is the one which is involved in the translocation. Once the translocation is over, what is happening, friends? Uncharged tRNA comes to the E side, it get exited and peptide bond also got cleaved out. Now friends, we have to separate the large ribosomal subunit with the small ribosomal subunit. That's why what is this particular ERF3 GTP will do? It immediately recruits the EIF5 also, and it immediately recruits the EIF6 also. Friends, whenever there is a presence of EIF5 and 6, immediately large ribosomal subunit can be dissociated with the small ribosomal subunit. Friends, entire complex can be dissociated. Friends, here comes a very interesting point. Once this entire thing got separated, friends, you people has to listen to me. Once again they will assembled. Friends, all of you just listen my statements. If you look into the prokaryotic organisms are only very small, and their polypeptides is also very small. You should not think that in case of a prokaryotic organism polypeptides are very long. No, they are very small. Then you only imagine in case of eukaryotes like us, you can take an especially human being also. In case of us, our polypeptides are very large polypeptides. There is no doubt about it. They are very large polypeptide. Now you only try to understand what exactly I am saying. Protein synthesis has to occur very fast. Listen to me carefully. Protein synthesis has to occur very fast. Second point, we are having the limited time. Synthesis has to occur very fast. Time is limited. The most significant point, the length of the polypeptide is too much. The length of the polypeptide is very long. So friends, if one ribosome complex is present, what friends? If one ribosome complex is present, it is not at all sufficient to synthesize the polypeptide. That is the reason, friends, in the eukaryotic organism, the mRNA is only one. But how many ribosomes can be associated? Many ribosomes. See, this is one ribosome complex, this is another ribosome, this is the another ribosome. And once again, most of the students will confuse that these ribosomes are basically synthesizing the different proteins. That is also absolutely wrong statement, friends. This particular ribosome synthesizes a polypeptide, here also polypeptide, here also polypeptide. All are same polypeptide only. So just how these two can be connected? Just by polypeptide. How it will be connected? Just by the polypeptide, nothing but by the peptide bond. By that, friends, all the students listen. Here many ribosomal complex are synthesizing the same protein only. As a result, friends, within the less time we can synthesize the long polypeptide. So one mRNA with multiple ribosome, what do you call it? This as, we will be calling this as the polysome complex. And every student has to remember, the polysomes are only found in the eukaryotic organism. But friends, when you look into the prokaryotic organism, what we remember, friends? In case of a prokaryotic organism, one mRNA and one ribosome complex only, nothing else is present. Here, in case of eukaryotes, mRNA is only one, but how many ribosome complexes are there? Many ribosome complexes are there. Don't forget that. And I don't know, friends, whether you remember my words or not. While discussing the RNA processing or while discussing the post-transcriptional processes, I have highlighted this point. What is the point I have highlighted? Friends, 5' capping and 3' polyadenylation. These two are required for the eukaryotic translation. That is what we studied actually. Now the same thing is happening, right? How does the ribosome complex enter inside? With the help of a, you can see, right, a capping complex. With the help of capping complex they enter inside. How does the ribosomal complex can be dissociated? With the help of a polyadenylation. So they will get dissociated, once again associated. They will get dissociated, associated. So like this, friends, mRNA is only one. Multiple ribosomes will be added continuously. This is what do you call polysomes. And polysomes never ever found where, friends? In case of prokaryotes. So friends, this is about the complete process of the translation in case of a prokaryotic organism also, friends, as well as, friends, eukaryotic organisms also. Kindly don't forget it. So next one, before going to the post-translational modifications, it is a very important, friends. We have to read compulsory the antibiotics that block the process of translation. Friends, whatever the antibiotics that I kept in this particular slide, they are not at all sufficient. So you people check after 11 or 11:30 a.m. in the morning today. I will upload one important PDF. Okay, so team will upload a PDF. In this particular PDF, I will keep the antibiotics are blocking the translation also, antibiotics blocking the transcription also, antibiotics are blocking the replication also. Friends, on an average, I want you people to remember at least 25 antibiotics, including the translation in prokaryotes, eukaryotes, transcription in prokaryotes, eukaryotes, replication in prokaryotes and eukaryotes. Every student listen to me carefully. When compared to the antibiotics blocking the replication, we should majorly study the antibiotics that are blocking the translation in prokaryotes and eukaryotes, antibiotics that are blocking the especially transcription. Both you have to remember, transcription, translation. This chart is not at all sufficient, friends. It's just an overview. Whatever the PDF that I will upload in the website, I want you to read it. There is no difficult concept over there, right, my friends? We have to read the simple concept only, na? I mean to say, you have to remember, here I mean to say you have to remember a lot of information. Name of the antibiotic, which class it belongs to you, which class it belongs, most important, friends, what kind of function it is doing. That is the only thing you people has to remember for the exam. An example, first of all, how can I define the word antibiotics? How can I define? Antibiotics are the drugs. Every students listen to me. Antibiotics are the drugs, or antibiotics are the chemical substances which are synthesized from the microorganisms and which is used to kill other type of microorganisms, which we can call antibiotics. Which means, listen friends, antibiotics are synthesized from the microorganisms only, no doubt about it. That antibiotics only used to destroy or kill the another group of microorganisms. That is what we can call antibiotics. The interesting point every student has to remember is that, friends, 90%, not 5 or 10%, friends. Everyone listen, every one of you. 90% of the antibiotics that we are using in our daily life are for a different kinds of diseases. 90% of the antibiotics that block the process of translation. 90% they can easily block the translation. And friends, not only antibiotics, friends, even certain toxins also, they can also block the process of translation. Okay, I am just explaining you the most important ones. You have to read everything in the PDF. Here very less to explain. This is the area you have to remember. Aminoglycosides. I want you to remember the best example: streptomycin, neomycin, gentamicin. These three. What is the majority or major function of the aminoglycoside, friends, or kanamycin or streptomycin? Peptide elongation at the bacterial 30S ribosomal subunit. I can tell you in a very simple way, friends, all these aminoglycosides, they can block. What they can block? You will read. They can block peptidyl transferase activity. Upon blocking the peptidyl transferase activity, it never allow the process of elongation, friends. As a result, protein synthesis will be abruptly stopped. Abruptly stopped means it stopped immediately in the middle. Abruptly stopped means stopped, which is stopped in the middle. If you want to remember the side effects, remember. Otherwise, do not worry at all. Next, friends, amphenicols. The best example of the amphenicols are what? Chloramphenicol and thiamphenicol. Chloramphenicol and thiamphenicol are the broad spectrum antibiotics, friends. Their function is what? Chloramphenicol is already asked in the exam. Their function is what? Same, friends. They are also blocking the peptidyl transferase enzyme, and at the same time, friends, chloramphenicol bind to the A site. We all people commonly know, right, friends? To the A site, who will come and attach us? To the A site, amino acyl tRNA will come and attach us. You only tell me, friends, do you think without tRNA any kind of a protein synthesis will occur? Highly impossible, right? So this chloramphenicol block the A site. By that, it can also stop the protein synthesis. Very simple. Macrolides, like only remember azithromycin, erythromycin. Remember these two. What is their function? So friends, here comes a very interesting point. Azithromycin or the erythromycin, they basically block the ribosomal translocation, which means every student has to listen. They are basically blocking the translocation factors. I hope every student remember. In case of a prokaryotic organism, what is the factor? EFG GTP. In case of eukaryotes, what is the factor? EF2 GTP. Is it? These two are the one which are involved in the translocation. Friends, what is the speciality of the erythromycin and azithromycin? The speciality is that they can act on the prokaryotic organism also, friends. They can act on the eukaryotic organism also. So they block this also and they block this also. By that, the protein synthesis can be inhibited in the prokaryotes and eukaryotes by the erythromycin and azithromycin. Kindly don't forget that. Then another very important ones are tetracyclines, which we commonly take for many kind of infections. Doxycycline, minocycline and tetracycline. Doxy, mino, tetra. Remember these three without fail. Okay. How it is blocking? It blocked the polypeptide synthesis by sterically blocking the recruitment of amino acyl tRNA at the A site. Same, whatever the function performed by the macrolides, what they have done? They block the translocation. Here what they are blocking? They are never ever yellowing the amino acid with tRNA to the A site. If no amino acid is associated with the A site, obviously, friends, there is no way that the peptide bond can be formed. When there is no peptide bond formation, how does the polypeptide synthesis can occur? Polypeptide synthesis cannot occur. This is how different type of antibiotics are involved in the process of translation blocking. As I told you, this information is not sufficient. I will keep the PDF on the website. So you people please try to strictly try to read that in the tomorrow class itself. I mean to say day after tomorrow molecular biology class. Undoubtedly I will ask you. I am requesting all of you, every student has to remember at least 4 antibiotics. Blocking the prokaryotic translation, eukaryotic translation. 4 antibiotics blocking prokaryotic transcription, eukaryotic transcription. 4 antibiotics blocking the prokaryotic replication and eukaryotic replication. I don't know what you do, but I want you people to remember all these without fail. Never ever forget this point. Okay. Okay. Friends, is that clear everyone? If you have any kind of a doubt in the eukaryotic translation, you can ask me. So friends, in the tomorrow morning class, we will finish the plant hormones and we will enter into the most important chapter, that is the secondary metabolites. So I told you once again, maximum 100% by 31st, the 2 chapters will get over. You have to write exam on either April 2nd or April 3rd on these 2 units. So please kindly prepare accordingly. Okay friends, thank you all. That's all for now. I'll meet you in the tomorrow morning class once again, or the multiple ribosomes recruit ATSM. Kulakarni, yes dear. All the multiple ribosomes can be associated at a time only. Yes, at a time. All at a time. Once they synthesize the protein, they will get dissociated. But their association is simultaneous at a time. Okay friends, bye-bye. Have a good day to all of you.